Now, how do I change the slides? Ah, there's fine. Everything is fine now.
Thank you,
Okay.
Martin.
So mRNA-based systemically delivered gene therapy using nanomaterials. This is the general introduction. Talk about nanomaterials. I abbreviate them as NMS. And the two nanomaterials I'll be talking about are exosomes that are also known as, in fact, more accurately, extracellular vesicles, or EVs, and they range in size from 30 to 100 nanometers. Lipid nanoparticles, LNPs, they're abbreviated as LNPs, and the size ranges from 50 to 300 nanometers, depending on how you make them, and you can control their size. And then I'll talk about their suitability for gene delivery. I'll talk about if mRNA is better or DNA, for what particular purposes, and how to load the mRNA into EVs, not an easy task. Making the nanomaterial directed, because, unless these things are directed, you cannot administer them systemically. And direct injection of nanomaterials into cancer or any other disease, is inefficient. Not all diseases, not all cancers are accessible for this kind of direct injection. And then I'll talk about our own work with directed EVs and prodrugs in treating cancer, and some examples of treatments using directed LNPs, and a brief mention of clinical trials that are in progress. So the specific targeting, as I just mentioned, is important because one is interested in systemic administration of gene encoding materials like nanomaterials. And it is made possible because receptors and ligands are overexpressed in many diseases. And examples are given in here. HER2 is overexpressed in HER2-positive breast cancer, and you can see that it is in a different color. The reason being that this is the one on which we have concentrated on, and I'll be discussing it in greater detail as we proceed. Then PSMA is overexpressed in prostate cancer, bombesin, and many others. And of course, you can see the list. It goes down, and not merely cancers, even in Alzheimer's disease. Cannabinoid CB2 receptor is overexpressed, and dopamine in Parkinson's disease. So let's talk about EVs for systemic gene delivery, their suitability, and some issues that they have. The big advantage is that EVs are made by every cell of our body, not only ours, animals, even bacteria make EVs. And so they tend to be non-immunogenic. They are really nature's own antigen delivery system by which cells communicate with each other, even at a distance. And tetraspenin CD9 mediated fusion of the EVs with the target cell results in rapid delivery quick delivery of the material, like mRNA, for instance. And therefore, there is rapid effect. They're small, as I just mentioned, and they therefore avoid clearance by the RE system. And they can extravasate through the vessel fenestrations that are found in tumors, in the vasculature of tumors. They avoid liver lung entrapment for the same reasons, and the resulting toxicity, avoid lysosomal endosomal pathway, and many of them, HEC293 dendritic cells, mesenchymal stem cell exosomes, they're non-toxic. And they can be made to target specific receptors and ligands by, for example, constructing fusion proteins consisting of a targeting domain and an EV anchor domain. So proteins that are on the EV surface, you combine them with a targeting domain. And two examples of anchor domains on EVs I've given here, LAMP2B and CD47. So what are the issues? What are the things that may be problematic? Well, they're easy to load with the small RNAs, and there have been very successful treatments delivering siRNA and things like that. But bigger molecules are difficult to insert. For example, mRNA, and there have been several attempts which were unsuccessful, but we have succeeded in loading EVs with mRNA of about 573 nucleotide length. And this was done for the first time. Everybody else had failed. We succeeded. But larger mRNA introduction would definitely be problematic. So this is their limitation, although still they are very useful. And one other thing is that so far they're difficult to make in large quantities, although a lot of progress is being made in upscaling them. And I've given an example here of different workers. For example, nitrogen cavitation with sudden release of pressure can increase the yield. Incubation with liposomes by the producer cells can increase yield as well. And then use of cross-linking reagents like formaldehyde and a reducing agent, dithiotol, can increase the yield. Now let's come to the LNPs for systemic gene delivery, and once again, the advantages and some issues. A big advantage, you can see the shape of an LNP, and you can see that the mRNA is safely ensconced inside them. And one big advantage is that they can enclose large molecules, very large mRNAs or anything else. And they're very easy to make. All you need is a lipid solution in ethanol. As far as mRNA is concerned, mRNA in ethanol, lipid solution in ethanol and mRNA in water using microfluidic devices. And this one gives you some details of how this can be done for smaller scale and large scale production. And as I mentioned, can deliver large molecules like mRNAs of various sizes. And then like EVs, they can be made to target specific receptors, ligands. And this can be done by pegylation, manipulating their chemical composition, or manipulating their charge. For example, if you combine them with the cationic lipid DOTAP, then depending on the amount of DOTAP, for example, 10 to 15% DOTAP containing LNPs target spleen, and those containing 50% of this lipid target lungs. And this is really quite specific, and there are many other such examples. Issues. Lipid components of which these LNPs are made can cause lung and liver injuries. And as I mentioned, pegylation is one way of making them directed, and in fact, it is very often used for this purpose. But it can stimulate the PEG, can stimulate the complement system, inducing hypersensitivity, and the consequent antigenic response can result in shortened circulation time, mitigating effectiveness. Now, of course, the countermeasures are being taken, and I think that both of these, the LNPs and EVs, really have a big future, and they hold a very important promise in enhancing gene-mediated treatment of diseases. So the countermeasures, as far as these problems of LNPs are concerned, is improving LNP or the lipid biocompatibility by use of improving LNP biocompatibility by use of biodegradable lipids. Attaching PEG molecules on LNP surface through labile bonds, sensitive to, for example, serum albumin, promote slow depegylation, manipulation of PEG surface density, chain length. So the idea is that you get the advantage of pegylation, but the PEGs doesn't stay for long enough to cause the problems. Next thing. So as you know, DNA is mainly used so far, although now things are changing with the COVID vaccine. In gene delivery, DNA is often used. Traditionally, that was used. But the question is, can RNA be better for some treatments, for example, cancer? There are, a few things to consider. As far as safety goes, DNA is less safe because it poses the risk of insertional mutagenesis, which can cause, undesirable effects, kind of mutations that you don't want. And this risk is not posed by mRNA. Then there is the speed, efficiency, and durability of expression. Now, DNA can generate several copies of mRNA, but it has to enter the nucleus for transcription, while mRNA is directly translated upon cytosol entry. So this part is 1+ for DNA, and this part is 1+ for mRNA. And another thing is that DNA enters the nucleus during mitosis, and its entry and gene expression are therefore delayed, especially in non-growing quiescent cells, because they are not undergoing mitosis. Okay, so how important are quiescent cells in cancer? We are talking about the fact that DNA may not be very suitable for delivering gene via DNA to treat cancer. And as I just mentioned, in non-growing cells, the DNA entry is inefficient because they're not undergoing mitosis. And so the question is, how important are quiescent cells in cancer? Okay, rapid growth of cancer cells make many nutrient deprived. They go away from the blood vessel, the vasculature of the tumor, and then they enter into quiescent phase. But this phase is reversible. They're not really dead. And so sooner or later, they revive and can, cause metastasis and recurrence of the cancer. Therefore, cancer cure requires killing also of non-growing cells. Now, there have been some direct studies to find what proportion of cells in a cancer may not be driving. So one example is the work of Shibamoto and Streffer. And what they did is to use cytochalasin B, which blocks cytoplasmic but not nuclear division. And they scored, multinucleate cells, total nuclei, cell number from which they could estimate how many cells are not multiplying. And they found that up to 67% of tumor cells can be quiescent. Okay, so clearly, quiescent cells are important in cancer. And the question is, how effective is mRNA versus DNA in delivering gene in quiescent cells? So Zhao et al. Examined this. They used a luciferase receptor gene transfection in these primary neuronal cortical cells, which do not grow, so non-growing cells. And with mRNA-mediated transfection of this reporter gene. They found expression within 1 hour, which peaked at 5 to 7 hours and ended at 12 hours. With DNA, no expression occurred until 7 hours, which was a bit delayed as we expected. But peak expression occurred at 36 to 48 hours, delayed, but it was an order of magnitude greater. And that can be understood because, as I just mentioned, one DNA molecule can generate several mRNA molecules. But if you're trying to kill the cells, this delayed higher expression really is irrelevant, because the kill cell, the dead cells, will have no expression. And so it would appear that mRNA would be better. And recent studies have confirmed this, even with using EVs. This is other people's work, and I won't go much into this, but here is a more clear example. And so Okamura et al., they examine treatment of malignant melanoma, implanted tumors in mice, and they delivered gene encoding Bex protein as DNA or as mRNA. And you can see that the growth of the tumor with DNA was much faster than was the growth of the tumor with mRNA, indicating that mRNA is more suitable. And the apoptotic index, which indicates the efficiency with which the apoptosis is taking place, was greater with mRNA gene delivery. So the conclusion, mRNA is better than DNA for gene delivery when killing of non-growing cells is important, as in cancer. It is also better when transient expression is needed to prevent non-specific effects, for example, in CRISPR-Cas involving therapy, which I'll discuss briefly a bit later. But DNA may be better when prolonged expression is required. For example, in cystic fibrosis, which it results because of the deficiency of CFTR protein, and you need production of CFTR protein on a prolonged basis to have effectiveness, to have effective treatment of cystic fibrosis. So now I will turn to our own work with directed EVs and prodrugs for treating cancer, and the approach that depends on gene-delivered prodrug therapy is, of course, referred to by that name and abbreviated as GDEPT. So this has been a highly collaborative effort. Some of the people are shown in here with their pictures, others have just their names. And you probably will have access to this slide, and you can take a look at who was involved. There are too many for me to mention right now. NIH people were involved as well, and this was supported by NIH U2 UH3 grants by Extracellular RNA Consortium, NCATS, to me, ACM. Okay, so these are our EVs, which we made from HEC293 cells using the kind of typical methodology of differential centrifugation, small, low G, a little bit higher, and then further higher G value. And this is the electron micrographs and nanosite characterization of their size, 3200, as I had mentioned. Transmission electron microscopy. And, the consortium of EVs has established that to be sure that a preparation is extracellular vesicle, you must show that they contain at least 3 proteins that are specific to EVs, and you can see that we have CD63, 81, MFGE8. So, they are bona fide EVs. Now, let me talk a little bit about prodrugs. Prodrugs by themselves are harmless, nearly completely harmless, but when activated by a viral or a bacterial enzyme, they become highly toxic. And if you can deliver the gene that encodes the activating enzyme, if you can deliver it specifically to cancer, then the capability to transform the harmless prodrug into a very toxic product would then be confined to the tumor, and you would then have a highly effective localized treatment without side effects. And also, because it's localized, you would need lower dose of the prodrug or any kind of drug. And so I've given you two examples here: genciclovir and thymidine kinase. It's a viral enzyme. And what happens is that this is genciclovir, which is quite harmless, but upon conversion or action of thymidine kinase from the virus, it can get converted into genciclovir triphosphate, which can muck up DNA synthesis. And this has been this is the only prodrug regimen that has been subjected to phase 3 clinical trials in glioblastoma multiforme patients, and craniotomy was used to inject the gene with a retrovirus containing the gene encoding thymidine kinase. But they found absolutely no benefit, so the trial was a complete failure. Another prodrug, CB1954, also called tretazekar, has also been subjected to clinical trials. It is activated by a nitroreductase from bacteria, Escherichia coli, for instance, and subjected to phase 1-2 clinical trials. But what it did was to establish safe dose of this prodrug, tretazekar, in humans. But once again, the results were equivocal. In fact, they didn't even warrant transfer to phase 3 trial. So we and others have identified what the issues were that led to this lack of success. And among them is that the wild-type enzyme that were used are not potent enough. There is insufficient gene delivery and expression. Viral vectors are problematic. The immunogenicity, which can prevent repeat dosage and possible pathogenicity. And then in neither of these, in fact, until we worked in this area, no attempt had been made to selectively target cancer. And as I mentioned to you, it is very important to do that, because not all cancers are accessible for direct injection, least of all cancer metastasis. And they had to administer it to the cancer site. So as I said, we have addressed these issues. No, as I'm saying now, we have addressed these issues, and this has been greatly helped by our discovery of a new prodrug called CNOB. Its chemical name is very complicated, and you can always look at our papers to find out what it is. And its drug, MCHB, again, a very complicated name, is very toxic, but a very important part is that it can be easily visualized in living mice by ordinary microscopy. And this is a detail of it. This is CNOB. This is the enzyme also which we discovered. It converts it into MCHB. And this is showing you that it is highly fluorescent, something like 15 times more fluorescent than the most fluorescent drug that is so far known, doxorubicin. And it can be the fluorescence can quantitatively reflect its potency, which is shown in here. The other thing is that it has a very, very high bystander effect. And what bystander effect is the ability of cells that can generate the drug, their ability to cause it to come out of them and affect neighboring cells and also kill them. And this is very important because no method of gene delivery can convert all the cells in a tumor or anywhere else into, confer on them the capability to generate the protein that you're interested in this case, the enzymes required for converting the prodrug into the toxic product. And as you can see, MCHB is of magenta color, and we were able to see if we can confine the activation of CNOB to the tumor, and this is the tumor, and you can see that magenta color is confined there and nowhere else. Okay, these are technical details to show, to determine how MCHB kills the cells. And, these are the various tests, and the conclusion is that it binds DNA, arrests cells in S phase, exposes phosphatidylserine on plasma membrane, sign of apoptosis, induces caspase 3, another sign of apoptosis, co-localizes with mitochondria, depolarizes mitochondria. So, conclusion, the MCHB causes mitochondria-mediated apoptosis and kills both growing and non-growing cells. And as I've been emphasizing, killing of non-growing cells is extremely important in effective treatment of cancer. Okay, so after we discovered this regimen, CNOB and CHRR enzyme, both of which we discovered in my laboratory, we then did a quick study in which, we were not interested at the time, it was very early in directing or anything, but only to determine how effective it was. So what we did was to implant tumors in mice that endogenously made the enzyme CHRR. And we found that indeed when, we used the cells, the tumor cells that engendered, that made the enzyme CHRR, the mice survived for, pronouncedly more effectively than the control ones. And then we also did, PK/PD studies using, all the Cmax and AUC 0 to 24, and using this well-known. And the conclusion is that, it is really quite effective. So, the initial indications showed that the regimen had promise and that it had very good PK/PD, characteristics. So we built upon these promising results by improving the enzyme, improving gene delivery, using directed gene delivery vehicles for systemic administration, as I've been emphasizing. So how we improve the enzyme? We used the standard techniques. One great thing is that, because of the magenta color of MCHB, we could easily determine which protocol, which changes, which approach engendered cells that had greater effectiveness for converting CNOB into MCHB simply by looking at the intensity of the magenta color. And you can see already that there are differences there. And what we did was to use error-prone PCR, DNA shuffling, and finally X-ray, and also a novel algorithm that was developed at Stanford, and crystallography. And the result is that we found that this HCHRR is a tetramer, and it became clear that if you strengthened the dimer-dimer interaction by appropriate changes in amino acids at the interface of the dimer-dimers, then you can improve the enzyme. And also another way is to enlarge the FMN. It's an FMN dependent enzyme. If you enlarge the capacity, the size of the FMN cavity, that too improves the enzyme. So, these kinds of amino acid substitutions, enhanced dimer-dimer interaction, and these kinds enhance the size of the, increase the size of the FMN cavity. And this way we were able to attain something like 100-fold greater activity of the enzyme. So this is a bit of a summary of what I've been talking about. MCHP causes mitochondria-mediated apoptosis, kills both growing and non-growing cells. It can be quantitatively visualized in living mice, which is very helpful for targeting. It has a very favorable PK/PD, the entire regimen, and MCHB has excellent bystander effect. And once again, you can see this is the tumor, and the magenta color is confined to the tumor alone, and this is the bystander effect. The yellow spots here show bacteria that are encoding the enzyme, and you can see only a few of them have this this yellow spot, and yet the entire monolayer is showing magenta color, indicating a very effective bystander effect. Okay, now the next thing is loading of exosomes with HCHRR6 mRNA, which proved to be a challenging task. Our initial attempts, and those of many other people, failed because of all sorts of things. Even if you were able to put mRNA into EVs by hooks or crooks, at the time of the transfer of the mRNA from EVs to the cell, they got, the mRNA got inactivated or just fragmented. But in collaboration with a private company, we were able to construct a unique plasmid, which employed the zip code, a sequence that was discovered at Harvard. And what they had found was that the mRNAs that are native to exosomes, as you know, the producer cells, when they make exosomes, those exosomes contain many of the contents of the producer cells, which include mRNA. So what they did was to look at, they sequenced mRNAs from many different exosomes and found that nearly all of the mRNAs found in these that were found in the exosomes contained a specific sequence, which was called the zip code. So we used that, and we were able to transfect, to make exosomes that contained HCHRR6 mRNA encoding HCHRR6 encoding mRNA and be effective. But the use of plasmid still entails use of DNA, which I've just showed you, can be highly inefficient, and plasmids can be harmful for patients. You never know what else they might do. So we now use in vitro transcribed HCHRR6 mRNA, which we call IVT, right? In vitro transcribed mRNA, and this proved more effective. And we call these EVs containing in vitro mRNA, in vitro generated mRNA, IVT exotapts, based on GTAPT terminology. And we'll discuss this further later. So now, we dispensed with the use of plasmids, as I just mentioned, because of the relative inefficiency, since it is still DNA, it has to get into the nucleus. And the other thing is the potential harmful effect of plasmids. So to enhance the prospect of clinical transfer, as I just mentioned, we are using IVT mRNA loading of EVs. And also, we use Tretazicar. And as I just showed you, which is CB1954, as I just showed you, the clinical trials with this have established a safe dose of this, which our own prodrug, although it is highly effective, does not have as yet. It has never been put in clinical trials. So this now is showing you that this figure here, that the introduction of in vitro synthesized mRNA into EVs requires many steps. And as you know, mRNA can be highly unstable. So it was essential for us to show that at each of these steps, the mRNA remained capable of encoding the protein that activates Tretazicar, or CB1954. And these are the technical details to show how we did the mRNA translation and all that. Appropriate plasmids were used. It was transcribed using this kit, another kit. Okay, and then this is important. The transcribed mRNA, that we arranged the transcription in such a way that it had 7-methyl guanosine cap at the 5' terminal, and poly A tail at the 3' end. This protects mRNA from degradation and facilitates its translation. Okay, so as I just mentioned, it is mRNA is subject to fragmentation, especially during EV-mediated delivery to cells. So as I said, it was necessary to ensure that HCHRR6 mRNA was functional in encoding a protein that would activate Tretazicar. Now, to do this directly with Tretazicar, whose toxic product we abbreviate as MPNQ. This is another very large name. But MPNQ, detection is very difficult. So what we did was to develop a color method, which made it much easier. And this is called the MCHB test. And I won't go into much detail of it. If you have any questions, I can then mention it. So all we needed to do was at every step, translate the mRNA into its encoded protein and see if it can develop the color that I just mentioned, which we refer to MCHB test. And if it developed the color, we knew that the mRNA was active. Okay, so this is the characteristics of IVT exosomes. HEC293 cells were transfected with mRNA using polyethylenamine. EVs were isolated, RNA extracted, functionality ascertained by MCHB test, quantified by qRT-PCR, the mRNA in the EVs, then the CT values, etc. You know, and what we wanted to see is how many copies did the EVs contain. And we found that the IVT, the in vitro transcribed mRNA-based EVs, contains something like 3.3 times 10 to the minus 2 copy per EV, whereas the plasmid had 2 times 10 to the minus 4 mRNA copy per EV, which was to be expected. As I mentioned, this plasmid requires entry into the nucleus, which is an inefficient process. And the consequence is that with the IVT EVs, you need less than 50 EVs to deliver one mRNA copy. With the plasmid one, you use 5,000. So it really was a big improvement. And we ensured that the mRNA was inside the EVs. Okay, so now we have EVs containing the mRNA. And the question is, how do you target it? And so far, we have worked with HER2-positive breast cancer, which is a pretty serious cancer. It is relatively under control, but there is still long-term issues with it. But in any case, we wanted a proof of concept, experiment or approach. And even though we have concentrated on HER2-positive breast cancer, the approach is generic and can be used to target any ligand by having an appropriate targeting moiety that can latch on to the exosomes. All right. So what we did was to make a HER2-positive receptor binding protein that is displayed by exosomes. This protein is called EVHB. We had an ML39 SCFV, anti-HER2 ML39 SCFV. As you know, SCFVs are small, highly specific antibodies, which we obtained from an appropriate plasmid, inserted into another plasmid. In any case, so the idea was to construct this protein, this EVHB protein. As you can see, it has a his tag for purification. And then it has this lactadherin C1, C2 domain, which can latch on to the exosome surface because it combines very strongly with phosphatidylserine, which exosomes express. And then what we did was with this plasmid stuff that I've been talking about, is to join it with this ML39 SCFV, anti-HER2 SCFV, and then the leader sequence in order to make sure that the protein would come to the surface of the EVs. So here it is. This is the exosome loaded with mRNA. We made a lot of this protein, purified it, and mixed it with the mRNA-loaded EVs, and then made these exodepts, loaded and targeted exodepts. By the way, this is the structure of predicted the structure of this EVHB protein. Now, these are very too many technical details, and I'll make it simple, or at least as simple as I can. We used ELISA to show that EVHB is needed to bind HER2 cells. Flow cytometry to show that exodepts bind only to HER2-positive cells. Actinomycin D and cyclohexamide to ensure that what the exodepts, when we did the experiment, what the exodepts transferred to the recipient cells, these HER2-positive recipient cells, leading to their death, was actually the SCHRR6 mRNA, because had it not been so, the results with these two reagents would be very different. And we get, the results are given in here, and you can look at them at your leisure. And if you have any questions, I'd be able to go into it. And it was clear that what killed the cells was the activated product of CB1954 made as a result of the fact that the mRNA, SCHRR6 mRNA that entered the recipient cells, HER2-positive recipient cells, then encoded the HCHRR6 protein, which converted CB1954 into MP and Q and killed the cells. So here are the main points. As I said, too many technical details, but SCHRR6 mRNA is inside the exosomes. I've already described that. It can generate MCHP fluorescence, indicating ability to activate reticular. That's the basis of the color reaction that I've been talking about, and I can go further into it if you're interested. Actinomycin D does not affect expression, which shows that what is delivered is not DNA, because this actinomycin D inhibits transcription, and had transcription been required and had DNA been involved, there would have been no killing. And therefore, mRNA is transcribed inside the cells into SCHRR6 protein, which then converts CB1954 into the activated drug that kills the cells. Okay, then further flow cytometry results, in which we wanted to see what the efficiency was with which EVs transferred mRNA into PT474 HER2. These are human HER2-positive breast cancer cells. And so we mixed known numbers of EVs containing known number of mRNA copies with known number of BT474 cells. And we found that even at 6 hours, each recipient cell received 5 copies when 10 copies were administered, indicating 50% transfer efficiency. And at later time, the efficiency was even greater. And what we attribute this is to the fact that the SCFV antibody that we use is really high. It has a very high affinity for HER2 positive, HER2 ligand, HER2 receptor. So it can bind very quickly and very tightly to the cells expressing the HER2 receptor, and resulting in this high efficiency that we saw. Now here comes the main thing of all this work. So what we did was to implant xenografts of BT474 human HER2 positive cells orthotopically into mice, and then administered the full regimen. And the black arrows show the times, the days on which we injected the IVT exodeps, and the sort of yellowish ones show when we injected CB1954 at the safe dose, which had been established, through these clinical trials, which I mentioned to you earlier. And I think what you need to concentrate on, to make it straightforward, is that all the controls, in all the controls, the tumor grew very lavishly, but in the mice where full regimen was administered, there was some initial growth, but by day 7, remember the last dose was of anything was delivered as day 7, but even after the last dose was delivered and nothing further was administered, the tumor, although it had some initial growth, then began to really not grow at all. Even for the next, let's see, 34 days or something. Yeah, suggesting total tumor death. Now we had to discontinue at this 34 days because of the protocol dictated by Stanford University. You can't prolong such experiments endlessly. And so, it's really turned out to be a highly effective approach, highly effective treatment. Okay, that's well and good. But how can we be so sure that there are no side effects? And so we did very extensive toxicology and histopathology analysis. It's very important because, EVs temporarily colonized pancreas, the spleen, and liver, and they could have easily delivered the HCHRR6 mRNA there, causing these organs to activate CB1954 and be hurt. This ML39 SCFV, which I've been admiring very much, and I think deservedly so, can also recognize mouse ERB2 expressing cells, which are not uncommon in mice. And so, the possibility was that Exodeath might have delivered the HCHRR6 mRNA also to normal tissue. And the question then was, did general toxicity accompanied the cancer? Treatment, I can say, really cured, because as you saw, no further growth occurred for several days after the treatment had been stopped. So we did many things, this hematology and serum chemistry, no toxic effects, organ histopathological evaluation, so many organs. Again, we found no deleterious effect. Hematology, no side effect. There were some minor changes in 3 components, but they were minor and transient, not indicative of bone marrow suppression. And also serum chemistry, slight increase in liver enzymes, but this is known effect of CB1954, and it is only transient. So there really is no side effects at all in a situation where the tumor was completely cured. Okay, so these experiments have been done in immune-compromised mice. But of course, immune-competent situation is the real thing. But we think that in immune-competent situation, the treatment might even be more effective. The reason is that the HER2 tumor ablation, which of course has taken place when we have cured the tumor in mice, it is known to evoke a strong anti-HER2 immune response, which would then definitely further help in preventing the growth, in destroying the tumor. And so our EVHP protein that I've just showed you, that enabled the exosomes or EVs to target the HER2 positive tumor, it has human components, and therefore an immune response is likely. But as I mentioned, the fact that we found no evidence of any general toxicity, that suggested very strongly that all the EVs were confined to the tumor. They may have stayed here and there for a while, but because of the strong affinity of SCFV antibody to HER2 receptor, they all went and got localized to the tumor. As a result, any immune response would target the tumor, and is likely, we think, to be even more effective in immune competent mice. And we are testing this in immune competent FVB-NG mice, which spontaneously develop HER2 positive breast cancer driven by an oncogenic form of human HER2, delta 16. EVs can cross the blood-brain barrier and also LNPs, and this is very important because, as you know, the metastasis of breast cancer and many others go to the brain, and obviously many brain cancers or diseases require treatment in the brain itself, so they can be used for that purpose as well. And as I've just mentioned earlier, the technology is generic. This is the kind of general representation of the EVHB protein. In the one that we used, the targeted ligand was the anti-HER2 scFv, but you can have any scFv or something else that targets PSMA, folate, or whatever, and treat other cancers. Now, so we are now preparing for clinical trials, and I won't belabor this point. They're all standard approaches, and yeah, so the details are described in these two slides, and we have worked out complete details. These are standard procedures dictated by FDA, and the slides are there. And if you like, you can go over the abbreviated version of it. Now, finally, I'll give you an example of a specific treatment with directed LNPs. This really is a very impressive work, not done by us, done by Rosenblum et al., 2020. And this employed CRISPR-Cas for gene manipulation. As you very likely know, Cas is an endonuclease. When directed by a guide RNA, it can cause double-stranded breaks in DNA at any intended site. And that break can be replaced with any desired DNA sequence by flanking it with sequences that are homologous to the DSP, the double-stranded region. Now, another thing is, this is kind of a side important thing to keep in mind. mRNA instead of DNA-based delivery of Cas is preferable for the following reasons: ease of making mRNA, its rapid expression, which is needed for CRISPR-Cas effectiveness, and equally important, the fact that the mRNA-mediated expression is transient, minimizes off-target cleavage. So what these people did was to inject Cas and mRNA and SG, the guide P10, and P10, by the way, is a tumor suppressor. They were co-loaded in DOTAP complex LNPs. As I had mentioned earlier, DOTAP is a cationic lipid, and its amount in the LNPs can determine its specificity as far as targeting organs is concerned, and they're referred to as SORT LNPs. So with 20% DOTAP, SORT LNPs containing these RNAs, etc., cause P10 editing in liver, but not in the spleen or lung. 50% SORT LNPs cause P10 editing only in the lungs. So it's really very impressive results, highly specific, so very promising. So that's the end of my talk. Thank you.